Hello, I am Vitali. I will briefly talk to you about our work entitled Exploring the Diversification of Plant Plasma Membrane Intrinsic Protein Channels from Now on PIP by Sequence Similarity Network Analysis. This network shows the complete major intrinsic protein superfamily, also known as aquaporins. It can be seen that the PIP subfamily is included in the major cluster together with classical or orthodox aquaporins. This group is characterized as being efficient channels for water. PIP are highly represented within plantae. About 10 to 30 PIP genes are encoded by embryophyte species, as can be seen in violet. They are found even in members of Asian plant lineages such as Elaginella and Cicomitrella. For years, efforts have been focused on elucidating whether this great multiplicity of isoforms implies diversity or functional overlap. Here we perform an analysis of the PIP subfamily diversity by sequence similarity network. Our first objective was to build an appropriate dataset to filter PIP aquaporin from MIP superfamily. The PIFAM database entry from UniProt was selected as a starting point. On the left, the schematic topology of PIP shows that they have a signature motif, the MPA, and a longer loop D compared to other aquaporins. This loop is involved in the particular gating mechanism triggered by calcium or pH that present PIP channels. These signatures were used to scan the BIP dataset to get the PIP subfamily subset by using the Bioset package in R software. We obtained a consensus loop D sequence by manual curation, and altogether, with the altered loop D of a Cocomixa PIP isoform, they were used to filter the MIP database. Based on this strategy, we obtained our dataset. Our second objective was to build a sequence similarity network using the EFI-ESD online tool and Cytoscape. In the following networks, each amino acid sequence is represented by a node. The PIP subfamily has a high amino acid identity, so the all-by-all pairwise comparison was performed with different stringent threshold values. We selected a threshold of 144. This network has a 3 cluster with different topologies, and it was chosen for further analysis. It can be seen here that this network has 3 big clusters, 9 minor clusters, and many singleton sequences. Well-known PIP aquaporins are indicated by arrows as a reference. The PIP1 paralog group is included in cluster 1, here in red. It is well documented that most PIP1 channels are unable to form functional channels unless co-expressed with PIP2 ones. This network has 3 big clusters with different neighborhood connectivity. Cluster 1 is an homogeneous cluster with high connectivity. On the other hand, despite so far no functional differences were reported, PIP2 paralogs are organized in several smaller clusters, each one with less homogeneous connectivity than cluster 1. The main difference between the 3 bigger clusters are on the length of N terminus, loop A, and C terminus, and their amino acid motifs. Then we analyze their correspondence among transport specificity reported for PIP in the literature and cluster organization in these networks. Color nodes are PIP with verified solute permeability, blank nodes are PIPs where no transport was detected, and gray nodes are non-tested PIPs. As can be seen, and with data obtained so far, solute permeability of PIP seems to be distributed in the different clusters without a preference. However, there are few results to conclude a clustering distribution bias. Then we analyze the organism distribution inside each cluster. The grasses PIP2 are not present in cluster 3, although we still cannot discriminate between the functional properties that characterize cluster 3. Its particular composition allows us to pose questions about when this group appeared and why it was not conserved within the grasses. To sum up, the PIP1 paralogs are in a homogeneous cluster with high neighborhood connectivity. PIP2 paralogs belongs to many smaller clusters with lower neighborhood connectivity. Length and different motif of N terminus, loop A, and C terminus discriminate between cluster 1, 2, and 3. PIP specificity for different solutes highlights the importance of each isoform independently of this clusterization, and PIP for dicotyledonous are broadly distributed in all clusters, while PIP2 grasses are absent in cluster 3. This work was funded by Agencia Nacional de Promoción de la Investigación y el Desarrollo Tecnológico y la Innovación and Universidad de Buenos Aires. Thank you very much.